Welcome to my channel. Here today I will be discussing about the size exclusion chromatography. By the name you can easily understand that it is one type of chromatography technique or process which is very essential in biology as well as chemistry, research and practical. So here I just will tell some overview of this process, like what is the basis of this technique or what is the chemical used in the stationary phase. So you can see that in the basis is the size and shape of the molecule which I want to separate, and the chemical nature of the porous gel is polyacrylamide gel, dextran and agarose. Here I have just written the principle, what you may read later. I just give the overall process or whole idea about the technique. And first of all, I just give the analogy. Suppose you have been given two types of ball, one large and one small, and you also have been given a small box. That small box only can accommodate a smaller ball. So you someone said you just you someone said you that you have to put the balls inside the box, and you try and see that only the smaller balls can enter into the small box, but larger balls cannot enter into the small box. So this same analogy is applied in this size exclusion chromatography. Here you can see the whole process. If you know little bit about the chromatography, then you are introduced to its picture. So here the chromatography column, and inside the column there is a beads, and here we can see that two types of molecule, like one large and one small. Large is the brown molecule and blue is the smaller molecules. So we these two types of molecules will be allowed to pass through the column, and then in the second picture we can see that brown molecules will move faster, but the smaller molecules, that is blue molecules, are moving little bit slower than brown molecules. Why? Because we I was saying before that the brown molecules are larger, so the beads cannot accommodate these brown molecules, but the beads can accommodate the blue molecules. So the blue molecules are moving slower, and in the third picture you can see that the brown molecules are coming out or elude faster or quickly, but the blue molecules are moving still now and are not eluded till now. And in the right side picture there is a bead which can accommodate the blue molecules but cannot accommodate the brown molecules. So by this picture you can understand the total basis of the whole process. So I again repeat that the bigger molecules will elute faster, which cannot enter into the beads, but the smaller ones which can enter into the beads will elute later. Here I will show a simple data analysis part with a picture. So I am drawing a graph, and here in the y-axis I just written the absorbance, because ultimately we have to determine the absorbance of the molecule. By this absorbance we can understand what type of molecule is that, and in the x-axis I just retain the time. So I was saying that the larger molecule will elute fast, so we will get a curve on lesser time, and in the in just later after that the elution of the larger molecule we will get the smaller molecules curve. Sorry. So it's a very bad picture, but you can understand the basis. So in the red curve is due to the larger molecule, and the small molecule gives the blue absorbance curve. And you can see the blue absorbance. This curve is showed in more time, and the red curve is showing in lesser time. Mainly, chromatography technique, all types of chromatography techniques are used to separate the molecules based on different parameters. And in this case, in size exclusion chromatography, the parameters is based on the size and shape. Suppose you have a biological cell, and as we know, in the biological cell there are different type of molecules or proteins, and you want to isolate a specific protein, and you know the molecular weight of the protein. So what you should do, you should choose the beads in the stationary phase according to the molecular weight of that your desired protein. So when you pass total protein extracts through the chromatography or column, then only that specific size of the proteins will get attached or get enter into the beads, and after those specific protein will elute. Thus you can isolate your specific type of protein. But yeah, you have to know the specific molecular weight of that molecule. Thank you for viewing this channel, and if you like this video, then please press like button and do subscribe.